Right. This is the second part of the chemistry review for human physiology. So I wanted to just finish up. We have this one should be shorter than the first recorded lecture. So if you are looking at time, this should be a little bit quicker. So we left off with ionization. So ionization, you basically are taking atoms and they're becoming ions. That means that they are either gaining or losing electrons. So it can happen with just a single atom, or you can have groups like carboxyl groups and amino groups that can be ionized. And they, like when you look at that, like here, your carboxyl group, the dash here means that it's attached to some other carbon containing chain usually. So here they're showing R is the whatever that group is attached to. So sometimes when it attaches, it can, here it's lost its one of the hydrogens. So you have a COO negative because it lost that the hydrogen, which is really contains that proton. So we can see that. And then here NH, NH2, the amino group became NH3 plus. So that's ammonium based, and it has a plus because it gained that hydrogen ion. So we can kind of see that happening. So those are with groups. So it can happen with single atoms, like we kind of saw when we were looking here at sodium giving an electron to chlorine, or chlorine accepting the electron from sodium. You create ions that way as well. So we have that. Free radicals. So free radicals are basically atoms that contain a single unpaired electron in its outer shell. And a lot of times when you hear about like beauty creams and stuff like that, they'll talk about, helps to fight against free radicals. Free radicals are highly reactive with other molecules, and that should be create another free radical due to the donor. So it should be another, now that I'm looking at that, sorry about that. And so, they can damage normal healthy tissue. And so, like I said, if you listen to ads that talk about like wrinkle cream and things like that, they'll talk about how it, fights the effects of free radicals. Free radicals, like I said, can damage healthy tissue. There are other components of the cell or cells themselves, like white blood cells, actually use free radicals to help kill bacteria. If you look at peroxisomes, they have free radicals in them that are used in decomposing things when we look at and breaking down components. So we can see that they're used sometimes for good, but like I said, they can damage healthy tissue, which makes them a problem as well. So antioxidants, so vitamin C, vitamin E, help to protect tissue. So like I said, if you've seen like a lot of times with face creams, they'll be like, free radical, works against damage, free radicals damaging effects and things like that. So these are things like the vitamin C and E can work to kind of counteract the effects of the free radicals. So we can kind of see that. So we've talked about covalent bonds, so polar and non-polar covalent bonds, and we talked about ionic bonds. So the last kind of bond we're going to talk about is hydrogen bonds. Hydrogen bonds are a weaker bond, and they don't make molecules. So a polar covalent, a non-polar covalent, and an ionic bond, all of those will make molecules. So you can make, sodium chloride. You can make water. You can make methane. Whatever, there's lots of different examples. They can actually make a molecule. Hydrogen bonds are weaker bonds, and they basically form in between molecules that are already made. So when we look at hydrogen bonds, they happen specifically in molecules that contain one of these three combinations. So if you have hydrogen that is directly bonded to oxygen, so like in water, where the oxygen is very electronegative compared to the hydrogen, and it creates those dipoles, the polar regions. You see it when you have hydrogen directly bonded to nitrogen. So that's another combination there. Sorry about my not so great handwriting with the computer. And then also the third one is hydrogen that is directly bonded to chlorine. So chlorine is at. So oxygen, nitrogen, fluorine are all very electronegative compounds. I don't know if you guys remember that from intro chem, but that means that they have a strong affinity, a strong liking for electrons. So when hydrogen, who is not very electronegative, comes in and it creates a bond with these, there is definitely a lot of very unequal sharing, and you create those very strong dipoles, and then you end up having attractions that form between those water molecules. So we'll kind of see that. I think that, do I show that? Yeah, I think in the next picture, I was like, with that. So these, when you see this, so hydrogen bonds don't make molecules. They basically, are an attraction between already formed molecules. So it makes, for substances that have hydrogen bonding in them, it makes their boiling points and things like that higher. So if you have if you were looking at a molecule of comparable size to water that didn't have hydrogen bonding going on, it would boil at a much lower temperature than water. So we can kind of see that. So, oh, there it is. So like when we look at this, I'm going to look at this and I'll go back to the page. So if we look at here, so here's our water molecule. We have, this is a polar covalent bond right here and right here. So polar covalent bond here that makes the water molecule itself. And since it's polar covalent, it creates a negative region around the oxygen, a positive region around the hydrogen because of that unequal sharing of the electrons. And then because this is negative and then this is positive, this green line represents, that's the hydrogen bond. It's basically the attraction of this hydrogen to that oxygen in these already made water molecules. So it's considered a weak bond because it doesn't hold molecules together, but it's an attraction of molecules. So like I said, it makes boiling points and surface tension stronger. A lot of the special properties of water come from the fact that it has these hydrogen bonds. So it's in the hydrogen bonds attraction between molecules. So in this case, water molecules. So if we go back one, it increases the surface tension, so water is attracted to itself. So if you have ever, like in chemistry, when you guys use a graduated cylinder and you would, fill it with water, when you read the amount of water in your graduated cylinder, you always had that meniscus. You guys remember the meniscus? So why does water form that meniscus? Because of that high surface tension, the water molecules stick to themselves and they stick to other things as well, like the inside of the graduated cylinder. So it forms that kind of like curve like that, and that's that tension there. So you have the attraction to itself. So the surface tension is higher. That makes things, makes it attracted to itself, and it increases boiling point. It does all those things. Hydrogen bonding is also important in molecules other than just water. So it's really important in structures like DNA and proteins. And we will see that when we look at protein synthesis, which is, we'll do biomolecules, and then I think it's the unit after that. Sometimes I have to think hard. I taught Gen Bio for a long time too, and sometimes the order is not the same. So the hydrogen bonding is used in DNA to help get that helical structure. We'll see that proteins, we're going to see that when proteins are made, they can go in alpha helixes or beta pleated sheets, and then those structures can fold in on themselves again. And how they fold and do that, one of the things that helps them fold in these very specific ways is the attraction of these hydrogen bonds. So we will be able to see that. So hydrogen bonding is really important for water and all the properties water has, but it's also very important in helping build the structure of DNA and proteins. So it's not just water that hydrogen bonding is important, but 9 times out of 10, when you see examples about hydrogen bonding, they usually use water because it's easy to draw and kind of see the structure of that, but also very important in DNA and protein, making those structures. Hydrogen bonding is involved in creating those specific shapes, which we'll find out is very important for both of these molecules. There are some other types of bonds that we'll kind of see with them. We have van der Waals, which previously was called London dispersion forces, which this type of bond it's a really weak bond. It's basically created when so remember how we talked about electrons? So you have the nucleus and you have those electrons that are moving around, and they don't just move in the little orbitals, they're moving in those kind of nebulous clouds. So what happens is sometimes when those electrons are moving around, they all kind of clump together at any given moment into one area. When that happens, you create a negative area, and then the area that doesn't have the electrons is more, doesn't have that negative charge. And so you can create these temporary dipoles. So the dipoles will essentially, you have this very fleeting, moment of negative that can allow things to interact with each other. But it's temporary because when those electrons move, then that van der Waals or London dispersion force is broken. You can see these around neutral molecules. So if you have a nonpolar covalent molecule, it can still have these London dispersion or van der Waals forces happening that can cause interactions to happen. And it helps to give 3D shape to large molecules. So you can kind of see that. If you would like, you can click on this link in your PowerPoint. I've lost my words, and watch that if you would like. I'm trying to make this, not too long. So here, this is showing myoglobin. So myoglobin helps, it's a molecule that you find in muscle tissues that helps to hold on to oxygen. So when we look at that, there are van der Waals forces that help to hold this in a very specific shape. So we're going to see when we start talking about protein synthesis, you have all these interactions that hold things in, like the helix here. This is the alpha helix, this kind of helical structure right here. Then this kind of looks kind of like bent or kinky kind of looking shape there, or like right here. Those are beta pleated sheets, and then this folds in on itself. Having this overall structure, van der Waals or London dispersion forces are help to create that overall structure of that large molecule. And within that, there's hydrogen bonding going on and things like that as well in here that help create this shape as well. They're just not showing all of those, but those are all happening there as well. So when we look at chemical reactions, like I said, you aren't going to have to balance chemical equations or do any stoichiometry or gas laws or anything like that. But you do have chemical reactions. So when you have a reaction, what's happening? So you could have things like synthesis reactions where bonds are forming. So you could take individual atoms and combine them together to get a new product, and that would be synthesis. You're creating something new. So that would be when bonds are forming. You could have different kinds of chemical reactions like decomposition, where you have bonds being broken. So if you had a larger molecule and it broke into 2 or 3 smaller molecules or individual atoms, that would be like a decomposition type of reaction. A lot of times we also see things like single or double displacement reactions, in which you could have a single atom or element with a molecule and they can swap partners, or you could have 2 compounds and they switch, where things are rearranged. So you're breaking and kind of building, so you're rearranging things within, like I said, maybe 2 compounds or an element and a compound, and that would be kind of rearrangement. So, and whenever you form, break, or rearrange different kinds of chemical compounds or elements, that's going to be an indication of chemical reactions. Like I said, you could have synthesis, decomposition, single replacement, double replacement. There's some specific ones out there, like you could have combustion, different things like that. There's lots of different kinds of chemical reactions. And we're going to talk about dehydration, reactions and things like that. We start looking at the organic molecules when we get to that next unit. All right, our next topic is solutions. So once again, you're not going to really have to do math with this. You can do math with solutions. You can find out molarities and stuff like that. We're going to talk about some very basics of that, but like I said, you shouldn't need a calculator at all on this test. So solutions are basically, they can be, you can have solutions of gases, solutions of liquids, solutions of solids, or like a liquid with mixing in a solid, but it's going to be a homogeneous mixture. So when you see the prefix homo, that's the same. Geneous is, so homogeneous together, that means, like where you dissolve something and it looks like a new solution, where you can't tell the difference between those components. So the solvent is the dissolving medium. Typically, especially in biology, when we talk about things, the solvent is typically water. Water's an excellent solvent. It's called the universal solvent because it is a polar molecule, and it will dissolve anything that is ionic and anything that is polar. Non-polar things don't mix in it. So if you put oil and water together, you get the two layers. You did not form a solution because they do not mix. But if you had salt and you put it in water, you stir it up, now you have a salt water solution. The thing that's being dissolved, or sometimes typically when we think of solution, we think of like, I take something solid and I put it into something liquid. But like you could take two liquids and mix them together. The liquid that's in the smaller amount is what's called the solute, or what's being dissolved if you had like a solid. So if you had, water and then you added sugar, the water would be the solvent, the sugar would be the solute, and then the water plus the sugar, so the solute plus the solvent equals the solution. They love to do that in biology. Everything likes to start with the same letter. So you have basically, solute plus solvent. Oh, that's really bigger than I wanted to be. Sorry about that. Oh. That looks like not equal. I want it to be equal. Solute plus solvent equals, there we go, solution. I promise in real life I have good handwriting. On the computer like this, it is not so great. But if you want to think about solute plus solvent equals solution. So those two things combined equal solution. And when we look at this, the solute is usually solid. The solvent is usually liquid when we typically think about it. But if you were mixing two liquids together, the one in smaller amount would be your solute and the one in larger amount would be the solvent, because it's like you're dissolving the smaller into the larger is the way that kind of that works for those. But typically we always think of when we're doing these, like we typically think of dissolving a solid into a liquid. And you can have solutions of gases too, and usually the same applies to like a liquid plus a liquid. Whatever gases in smaller amounts would be the solute, and whatever ones in larger amounts would be the solvent, and then all of it together would be that solution. When we look at percent solution, if you have percent solution is in parts per 100. So whenever you see percent, just like if you're figuring out your grade, it's parts per 100. So if you're doing percent solution, if you had, you would take the amount of your solute and then of your total solution times 100, and that's going to be the percent of your solution. So whenever you see percent, you want to think 100, just like when you do your grade. If you were trying to figure out what's my percent in this class, if you have the points that you have received out of the total points times 100, that equals your percent. So percent solution is parts per 100. When we talk about molarity is really another kind of term for concentration. So molarity is always moles per liter. So I don't know if you guys remember like Avogadros number and all that stuff. Like I said, you won't have to do math, but it's the moles per liter. So moles per liter is molarity, and molarity is big M, capital M, that's moles per liter. If you, like I said, I'm not going to make you do the math with us, but you, in intro chem, I'm sure you guys did molarity, in which, like, if they gave you, you know, if you have, you know, if they gave you a certain number of grams, like if you had a solution, what's the molarity of a salt solution that had 4.5 grams of sodium chloride with, 1 liter of solution. So if you were trying to find that molarity, you'd have to convert your grams to moles using your molar mass that you would get from your periodic table. And then you divide that by the number of liters, and that's going to give you the molarity. So it's always moles per liter. Alternately, you could say moles per 1,000 milliliters if they gave you a milliliter value with that, but moles per liter, and that gives you kind of the concentration of that. I don't have any, you don't have to do any math, like, so if you, when you go to do the exam, you won't have to calculate molar mass and then, do all that. So don't get too worked up, just moles per liter. And like I said, it's going to be the concentration. So you're going to take the molecular weight, which is the sum of all those atomic weights in grams. So like if you had glucose, you would have to take 12.01 is the atomic weight of carbon. So you take 12.01 times 6. Hydrogen is 1.01 times 12 because there are 12 hydrogens. And then oxygen is 16, so 16 times 6, and then you would add all those things together, and that would give you the mole, that gives you the grams per mole of glucose. So when we look at that, and then when we look at the molarity, that gives you that would give, you would be able to get the moles per liter there. So we have that. So if you have a chemical formula like this, so this is glucose, C6H12O6, we have lab. This week we're going to see, or depending on when you listen to this, the first lab that we come to, we're going to talk about glucose. When you look at a chemical formula like this is telling you what elements are in there. So we have carbon, hydrogen, and oxygen. Here we have 6 carbon, we have 12 hydrogens, and then 6 oxygen. So if you were asked what's in, how many atoms of oxygen are there, you can tell just by looking at the chemical formula what's in this, and then how much of each of those different elements that are in that particular molecule. All right, so Avogadros number. Avogadros number is 6.02 times 10 to the 23rd molecules. You can use that to for molar solutions. Basically, a mole of any substance or any solution contains the same number of molecules per liter. So if you were looking at, if you had so many grams, like if you had 4.5 grams of sodium chloride, you can convert your grams to moles, and then you could look at the number of molecules, because you would take the fact that Avogadros number, in 1 mole of any solution, there's 6.02 times 10 to the 23rd molecules, so you'd end up multiplying that, and that would tell you how many, molecules you had of the sodium chloride. Or you could use it for water, or you could use it for atoms. It works for all of those, but that's Avogadros number. I don't know. Should be a apostrophe there. We have that. So, and like I said, I'm sure that in intro chem, maybe stuff that you are like, oh, I didn't like it. I tried to forget that as quickly as possible. But like I said, I don't have unit conversion type questions on this, but I do want you to understand what these concepts are. When we look at osmolar solutions, basically, like if you have something that has is a salt or an acid, when you put it into solution, when you put it into water, it will break into the number of particles that are in that particular molecule or acid. So like if you had, you know, a salt solution, so like sodium chloride. So if you put sodium chloride, NaCl, into water, it's going to break into sodium, it's going to break into chlorine. So you have, when it goes into solution, you have twice as many particles as you would before it went into solution, because it dissociates once it goes into water. Or if you had like magnesium chloride, magnesium is a +2, chlorine is a -1, so it's the chemical formula for that would be MgCl2. So if you put magnesium chloride into solution and it dissociates, you get 1 magnesium and 2 chlorine, so that's 3 particles. So if you have basically something that's ionic, like a salt, so ionic, so a metal with a nonmetal, that dissociates into its however many components you have in that, or an acid. So if you had something like hydrochloric acid, like HCl, when that goes into water, it's going to break into hydrogen and chlorine. So it will break apart. If you have something that is made by covalently, it's made covalently bonded is how it's made. So like glucose. Glucose is made of covalent bonds. So the orbitals overlap and they share electrons. When you put glucose into solution, if you put a cube of sugar like glucose into the water, yes, it's gonna just like dissolve, but the actual, like when you get down to like molecule C6H12O6, that stays together. So it doesn't dissociate into those individual components. It would stay the same. So you know, like I said, you can break it out of like the cube shape, but ultimately the actual molecule of glucose, the C6H12O6, stays together. It doesn't break into hydrogen and carbon and oxygen individually. So you don't have to multiply by all those numbers. So here, the number of particles determines the osmolarity. So here's that NaCl. If you had 58 grams per liter, and you had 1 mole of it, it's considered 2 osmolar solution, because when this goes into solution, it breaks into sodium into a chloride ion. So you would end up taking your value and multiplying that by 2 when you look at those. So because it breaks apart. If you had, like I said, if this was like glucose, like C6H12O6, in 1 mole, it would still be 1 osmolar, because it wouldn't break down into individual ions. Oh, that's it. I was like, all right, so there is a study guide for you guys to basically use to help you kind of know what I'm looking for. Like I said, there's also the chemistry book that you can get in the library. Like I said, on the resources page of the MyMac page, it lists a whole bunch of page numbers in there. When you look at the page numbers, you're like, oh my gosh, there's so many pages. But when you start looking at what's on a page in here, it's not reading intensive. It's like, it's really just a workbook where you can go through. It gives you a lot of pictures, things to answer. Answers to those are right to the side there. So you can just kind of go through and review and look at those and make sure you understand, you know, what is an ion, what, you know, like what do different things mean, you know. And so this is available for you guys to check out. I don't have, usually I only teach 3 sections of physiology in the semester. I have twice as many as that. So there isn't enough of these for every individual person. So if you use it and you take the test and you're done with it, please make sure that you take that back so that somebody else can check it out. If your chemistry is very, very strong and you don't need this, you don't have to get it. It's just that it kind of just gives you a review of the basics of chemistry as how it's involved in kind of a biological science class. Like I said, it's not, there's no math heavy other than like subtracting, like if you're trying to figure out the number of neutrons, taking that, you know, your mass, your, you know, looking at the atomic mass, and then or yeah, the mass number, atomic mass number, and then taking your you know, the atomic number and doing subtraction, that's the worst math there is. Like I said, you're not going to have to use stoichiometry. You're not going to have to balance chemical equations. Like I said, I want you to be familiar with certain chemical symbols, but you don't have to memorize all the atomic numbers. You don't need to memorize all the you know, the symbols either. It's not meant to be like that. It's just to review so that, you know, like I said, if it's been a minute since you've done chemistry, you have a refresher, just so that when I talk about what's an ion, you know that that's a charged particle. You know, how does it become charged? It lost or gained electrons. Like, so some basic information like that. It's just meant to refresh you guys on that. So I'm hoping that it will be a test that you get a fairly good grade on, because like I said, you can use your book, you can use your notes, you know, and if you have questions, you can let me know, send me an email or ask, you know, during lab, after lab, something like that, office hours, anything like that. So I hope that you guys, I don't like how close I am. Hopefully that helps and it gives you guys an idea of, you know, some of the stuff that you may have forgotten in, you know, since you had to do this in intro to chem. But just let me know if you have questions. Have a good day.